Welcome to our comprehensive guide on base editing, a groundbreaking advancement in the field of gene editing. In this video, we'll explore what base editing is, how it works, and why it's considered a revolutionary tool for scientists and medical professionals. We'll also look at its potential applications, the technology behind it, and the ethical considerations that come with such powerful capabilities. Whether you're a student, a researcher, or just curious about the latest in biotechnology, this video will provide you with a clear understanding of base editing and its significance in modern science. Let's begin our journey into the world of precise genetic modifications. Base editing is a precise form of gene editing that allows scientists to directly convert one DNA base into another without causing double-stranded breaks. Unlike traditional CRISPR methods, which cut the DNA, base editing uses engineered proteins to chemically modify specific bases. This technology enables targeted changes at the single-letter level, making it possible to correct point mutations responsible for many genetic diseases. The process is highly efficient and reduces the risk of unwanted genetic alterations, offering a safer alternative for therapeutic applications. Understanding this technology opens new doors in genetics and medicine. To appreciate base editing, it's important to understand the basics of DNA. DNA is made up of four chemical bases: adenine A, cytosine C, guanine G, and thymine T. These bases pair up in specific ways to form the genetic code that determines how our bodies function. Even a single change in this code can lead to significant effects, sometimes causing genetic disorders. Base editing focuses on correcting these single-letter mistakes, offering hope for treating a wide range of diseases at their genetic roots. For the advent of base editing, traditional gene editing methods like CRISPR-Cas9 were used to modify DNA. These techniques involve creating double-stranded breaks in the DNA, which the cell then repairs. While effective, this approach can lead to unintended mutations or large-scale genetic changes. The risk of off-target effects has been a major concern, especially for therapeutic uses. Base editing was developed to address these limitations by enabling precise, single-base changes without breaking the DNA strands. Base editing uses a modified version of the CRISPR system, combining a catalytically impaired Cas protein with a deaminase enzyme. The Cas protein guides the complex to a specific DNA sequence, while the deaminase chemically converts one base into another. For example, cytosine can be changed to thymine or adenine to guanine. This process is highly targeted and does not require cutting the DNA, making it both efficient and less likely to cause unintended genetic changes. There are two main types of base editors: cytosine base editors, CBEs, and adenine base editors, ABEs. CBEs convert cytosine C to thymine T, while ABEs convert adenine A to guanine G. Each type is designed to target specific mutations, expanding the range of genetic diseases that can be addressed. Researchers continue to develop new variants to improve efficiency, specificity, and versatility, making base editing an ever-evolving field. One of the key advantages of base editing is its precision. By targeting single bases, scientists can make highly specific changes without affecting the surrounding DNA. This reduces the risk of off-target effects and increases the efficiency of the editing process. As a result, base editing is considered a safer and more reliable method for correcting genetic mutations, especially in clinical settings where accuracy is paramount. Base editing holds tremendous promise for medicine. It can be used to correct point mutations that cause genetic diseases such as sickle cell anemia, cystic fibrosis, and certain types of cancer. By repairing these mutations at the DNA level, base editing offers the potential for permanent cures. Clinical trials are underway to test the safety and effectiveness of this technology in treating a variety of inherited disorders, bringing hope to patients and families worldwide. Beyond medicine, base editing is transforming agriculture. Scientists are using this technology to develop crops with improved traits, such as disease resistance, higher yields, and better nutritional profiles. By making precise genetic changes, researchers can create plants that are better suited to withstand environmental challenges and meet the growing demands for food. This approach is faster and more targeted than traditional breeding methods, paving the way for a new era in sustainable agriculture. Base editing is also being applied in animal research. Scientists are exploring ways to correct genetic defects in livestock and pets, improve disease resistance, and enhance desirable traits. These advances could lead to healthier animals, more efficient food production, and new models for studying human diseases. The ability to make precise genetic changes in animals opens up exciting possibilities for both agriculture and biomedical research. Both base editing and CRISPR are powerful gene editing tools. They have important differences. CRISPR typically creates double-stranded breaks in DNA, which can lead to unintended mutations. Base editing, on the other hand, makes single-base changes without cutting the DNA. This reduces the risk of errors and makes base editing more suitable for applications where precision is critical. Understanding these differences helps researchers choose the right tool for each genetic challenge. Guide RNA plays a crucial role in base editing by directing the editing complex to the exact location in the genome where the change is needed. The guide RNA is designed to match the target DNA sequence, ensuring that the base editor acts only at the intended site. This specificity is key to the accuracy and safety of base editing, minimizing the risk of off-target effects and unwanted genetic changes. Creating an effective base editor requires careful engineering of both the Cas protein and the deaminase enzyme. Scientists modify these components to improve their activity, specificity, and compatibility with different cell types. Ongoing research focuses on optimizing these tools to expand their range of applications and reduce potential side effects. The engineering process is a testament to the ingenuity and creativity driving advances in genetic technology. Delivering base editors into cells is a critical step in the editing process. Various methods are used, including viral vectors, nanoparticles, and direct injection. Each method has its advantages and challenges, depending on the target tissue and the desired outcome. Researchers are constantly developing new delivery systems to improve efficiency, reduce immune responses, and ensure that the base editor reaches its intended destination within the body. Efficiency of base editing depends on several factors, including the design of the guide RNA, the activity of the base editor, and the delivery method. High editing efficiency is essential for therapeutic applications, where a large proportion of cells must be corrected to achieve a clinical benefit. Researchers measure and optimize efficiency through rigorous testing, ensuring that base editing can meet the demands of real-world applications. One of the main goals in base editing research is to minimize off-target effects, unintended changes to the genome. Scientists use advanced screening techniques to detect and reduce these effects, ensuring that the base editor acts only at the desired site. Improvements in guide RNA design and enzyme engineering have significantly lowered the risk of off-target mutations, making base editing a safer option for genetic therapies. Great power comes great responsibility. The ability to edit genes at the base level raises important ethical questions. Issues such as consent, equity, and the potential for unintended consequences must be carefully considered. Scientists, ethicists, and policymakers are working together to develop guidelines and regulations that ensure base editing is used safely and responsibly, balancing innovation with societal values. Regulatory environment for base editing is evolving as the technology advances. Different countries have varying rules regarding the use of gene editing in research, medicine, and agriculture. Regulatory agencies are tasked with evaluating the safety, efficacy, and ethical implications of base editing, ensuring that new therapies and products meet rigorous standards before reaching the public. Public perception plays a significant role in the adoption of base editing technologies. While many people are excited about the potential benefits, others have concerns about safety, ethics, and unintended consequences. Open communication and education are essential to build trust and ensure that the public understands both the risks and rewards of base editing. Scientists are engaging with communities to address questions and foster informed discussion. One of the most promising applications of base editing is in the treatment of sickle cell disease. This genetic disorder is caused by a single base mutation in the hemoglobin gene. Researchers have used base editing to correct this mutation in laboratory settings, restoring normal hemoglobin production. Clinical trials are underway to test the safety and effectiveness of this approach in patients, offering hope for a long-term cure. Cystic fibrosis is another genetic disease that could benefit from base editing. Caused by specific point mutations in the CFTR gene, this condition leads to severe respiratory and digestive problems. Scientists are developing base editing strategies to correct these mutations, with the goal of restoring normal gene function. Early results are promising, and ongoing research aims to bring these therapies to patients in the near future. Base editing is also being explored as a tool for cancer research and therapy. By correcting mutations that drive cancer growth, scientists hope to develop targeted treatments that are more effective and less toxic than traditional therapies. Base editing can also be used to engineer immune cells to better recognize and attack cancer cells, opening up new avenues for immunotherapy. Inherited eye diseases, such as retinitis pigmentosa and Leber congenital amaurosis, are caused by point mutations in specific genes. Base editing offers a way to correct these mutations and potentially restore vision. Researchers are testing base editing in animal models and early-stage clinical trials, with the hope of developing lasting treatments for patients with genetic vision loss. Base editing is just one of many tools in the gene editing toolbox. Scientists are developing new technologies such as prime editing that offer even greater precision and versatility. By combining different approaches, researchers can tackle a wider range of genetic challenges and develop more effective therapies. The field of gene editing is rapidly evolving, with base editing playing a central role in this exciting progress. Despite its promise, base editing faces several challenges. These include improving efficiency, reducing off-target effects, and developing safe and effective delivery methods. Researchers are also working to expand the range of base changes that can be made and to better understand the long-term effects of editing. Overcoming these challenges will be key to realizing the full potential of base editing in medicine and beyond. Future of base editing is bright. Ongoing research aims to make the technology more efficient, versatile, and accessible. Scientists are exploring new applications in medicine, agriculture, and environmental science. As our understanding of genetics deepens, base editing will play an increasingly important role in shaping the future of health and society. The possibilities are limited only by our imagination and commitment to responsible innovation. Collaboration is essential for advancing base editing. Scientists from diverse fields, biology, chemistry, engineering, and medicine, are working together to solve complex problems. International partnerships and open sharing of data accelerate progress and ensure that discoveries benefit people around the world. The spirit of collaboration is driving the rapid development of base editing technologies. As base editing becomes more widely used, education and training are increasingly important. Scientists, clinicians, and students need to understand the principles and techniques of base editing to apply it effectively and safely. Universities and research institutions are developing specialized courses and workshops to build expertise in this cutting-edge field, ensuring a new generation of skilled professionals. Base editing frequently makes headlines as new breakthroughs are announced. From successful disease treatments to advances in crop science, the technology is capturing the public's imagination. Media coverage helps raise awareness and spark interest in genetics, encouraging more people to learn about and engage with this exciting area of science. Behind every scientific breakthrough are real people whose lives are changed by new technologies. Patient stories highlight the impact of base editing, from children cured of genetic diseases to families given new hope. These stories remind us of the human side of science and the importance of continuing to push the boundaries of what's possible. Funding is a critical factor in the development of base editing technologies. Government agencies, private foundations, and industry partners all contribute resources to support research and development. Adequate funding ensures that scientists have the tools and support they need to make new discoveries and translate them into real-world applications. The open science movement is transforming how research is conducted and shared. By making data, methods, and results freely available, scientists can build on each other's work and accelerate progress. Open science fosters transparency, collaboration, and innovation, ensuring that the benefits of base editing are accessible to all. Intellectual property rights play an important role in the development and commercialization of base editing technologies. Patents protect innovations and incentivize investment, but they can also create barriers to access. Balancing the interests of inventors, companies, and the public is a complex challenge that requires thoughtful policies and international cooperation. Base editing has the potential to impact people around the world. From treating rare diseases to improving food security, the technology offers solutions to some of humanity's greatest challenges. International collaboration and equitable access are essential to ensure that the benefits of base editing reach everyone, regardless of geography or income. Base editing is not limited to medicine and agriculture. Scientists are exploring ways to use the technology to address environmental challenges, such as restoring endangered species, controlling invasive pests, and cleaning up pollution. These innovative applications demonstrate the versatility of base editing and its potential to contribute to a healthier planet. Ensuring the safety of base editing is a top priority. Researchers follow strict protocols to test new technologies in the lab and in clinical trials. These protocols include extensive screening for off-target effects, monitoring for unintended consequences, and adhering to ethical guidelines. Safety is at the heart of every step in the development and application of base editing. Engaging with communities is essential for the responsible development of base editing. Scientists are working to involve patients, families, and the public in discussions about the risks and benefits of the technology. Community engagement helps build trust, ensures that diverse perspectives are considered, and guides the ethical use of base editing in society. Base editing is becoming a key topic in science education. Schools and universities are incorporating lessons on gene editing into their curricula, helping students understand the science and its implications. Educational outreach programs and interactive resources are making complex concepts accessible to learners of all ages, inspiring the next generation of scientists. Women are making significant contributions to the field of base editing. From leading research teams to developing new technologies, female scientists are at the forefront of innovation. Efforts to promote diversity and inclusion are helping to ensure that the field benefits from a wide range of perspectives and talents. Young scientists and students are driving innovation in base editing. Through competitions, internships, and research programs, youth are gaining hands-on experience and contributing fresh ideas to the field. Their enthusiasm and creativity are shaping the future of gene editing and inspiring others to get involved. Growing number of startups are focused on developing base editing technologies for medical, agricultural, and industrial applications. These companies are translating scientific discoveries into real-world products and therapies, driving economic growth and creating new opportunities for innovation. The startup ecosystem is an important engine for progress in the field. Conferences and scientific meetings provide opportunities for researchers to share their latest findings, discuss challenges, and build collaborations. These events are essential for advancing the field, fostering new ideas, and keeping the scientific community up to date on the latest developments in base editing. Data is at the core of base editing research. Scientists rely on advanced analytics and bioinformatics to design experiments, interpret results, and identify new targets for editing. The ability to collect, analyze, and share large datasets is accelerating progress and enabling more precise and effective gene editing strategies. As we look to the future, base editing stands as a symbol of scientific progress and human ingenuity. The technology has already made remarkable strides, and its potential continues to grow. By working together, embracing innovation, and upholding ethical standards, we can harness the power of base editing to improve lives and shape a better world for generations to come. Summary: base editing represents a major leap forward in gene editing technology. Its precision, efficiency, and versatility make it a powerful tool for addressing genetic diseases, improving agriculture, and tackling environmental challenges. As research continues and new applications emerge, base editing will play an increasingly important role in science and society. Thank you for joining us on this journey through the world of base editing. Stay curious and keep exploring the frontiers of genetic innovation.